So let's return to secondary structure prediction. And let's begin with something simple. Why do we predict secondary structure? So the lecture is about protein structure prediction. What role plays secondary structure in this game? We want to predict 3D structure because 3D structure relates to function. Why do I at all talk about secondary structure prediction? It's an intermediate layer, yes. This is one way of putting it. Another way of putting it is, again, the protein folds independently to a unique three-dimensional structure, but we cannot predict the way it looks. So we want to predict protein structure for all proteins in, say, human. We have about 20,000, but we cannot. We can do it through comparative modeling. Comparative modeling means that I have an experimental structure, I have a sequence for which I want to know the structure, and I can map that sequence to that experimental structure, and I simply copy the coordinates. If I find that the sequence identity between the two, the similarity is high enough, then I can copy the coordinates, and that's comparative modeling, and that I can apply to round about half of all the proteins, ballpark. For the other half, some of those we can predict contacts, and typically in this lecture we get to that. This year we may not get to that point of contacts, but the fraction for which I can use contact predictions, 2D predictions, to really get to the structure is again small, in the hundreds. For all the remainder, roughly 10,000, I cannot do 2D and I cannot do 3D. So I have to revert to some simplification, as you correctly called it, an intermediate layer, something simpler. Now, the next question is, why do I care? So, okay, I do secondary structure prediction because I can. But why would I want it? Why is secondary structure, helices and strands, why are they relevant? Does every protein have regular secondary structure? Where regular secondary structure I call helices and strands. Now you remember the third state, if you want to call it state, I refer to as other. So, does every protein have either helices or strand or both? If you answered no to that, so if you imagine there would be many proteins without any regular secondary structure, then there really wouldn't be a point to secondary structure prediction, right? Then I simply say everything is the other state, and in a certain case we will talk about proteins that are like that. But most proteins, in fact, have regular secondary structure. For most proteins, the content of regular secondary structure is about half. So half of the protein is either helix or strand. Do you remember how secondary structure is essentially described, or what stabilizes secondary structure? Chemically, or biophysically, or biochemically, whichever word you use for that. I'm talking about the kind of bond that stabilizes secondary structure. Hydrogen bonds. Hydrogen bonds are relatively weak on the scale of all the forces that hold together proteins and that make them fold. Hydrogen bonds are relatively weak, but secondary structure is stabilized by many of them. Now, the big difference between secondary structure, helices and strand, is in helices the bonding is local, typically between residue I and I+4. So if you look at a window of, say, 5 consecutive residues, the first and the last bind, or have a hydrogen bond in between. While strands come together from far apart in sequence often. So the two residues that bind to each other may not be within a 5 or 10 residue window. In that sense, strands, or the moment strands are packed together or bind, they're called sheets, and sheets are therefore often called the non-local secondary structure. Secondary structure formation is supposed or thought to help in speeding up folding, and it is clearly something that tells us something about proteins and function. There are, for instance, 2 helices that bind into the DNA groove. And when you see this 2 helix motif, that's one of the motifs binding DNA. So if you find that motif, then that already tells you something about function. Now, and it tells you a little bit about comparing proteins. It tells you a little bit more than sequence alone. We talked about very simple secondary structure prediction methods that essentially look at probability. So the probability of a particular residue to be in either of the 3 secondary structure states, and then to assign the highest probability to every single residue and use that as a secondary structure prediction method. We advanced over that, again with a probability method that looks at a window. So saying that the central residue is a particular amino acid, such as a proline, what's the probability of it breaking a helix if its environment is, if next to it is a leucine, or something like that. And I told you that these methods for many years were thought to not reach accuracies much higher than 60-something%. The official number was 65. It turned out that was slightly overstatement of the day, but clearly they could not reach 100%. And I told you that the argument for that was that some formation of secondary structure is global. So whenever you look at a consecutive window of 5, 10 residues, not all the contexts that form the secondary structure are in there. This, in fact, is exactly the reason why strands have level of accuracy that is almost random, smaller than 40%. Because sheet formation, strands coming together, is non-local. So we discussed the option of making the window longer. So the most extreme end of it, we could make the window as long as the entire protein. That doesn't work because then we could predict secondary structure only for proteins of 102 residues, or whatever the number of the first one is, and proteins have very different lengths, have an amazing distribution. So instead I could argue, okay, proteins are made of domains, let's pick a typical domain length, 100 residues, and let's run the window of 100 residues and predict secondary structure for 100. That should contain all the information. Yes, but the problem is that when you make the window too large, you add too much noise. So the signal is in there, but it gets lost in an ocean of noise. So that doesn't work either. And we had this problem, I said that segments that are predicted are too short. I introduced the concept of neural networks as one example of machine learning devices as a means to predict secondary structure. Now, in a neural network, you have input units and you have output units. We want to predict secondary structure in 3 states. Can somebody tell me how you would design the output units of a neural network to do that? How many output units would you use? The answer is really trivial. Just dare to give it. You look like you have it. Yeah, that was the easy one. But how do you do the input? So you want to code for a window of consecutive residues. Say 5. How many input units are those? So the answer this time is not 5. Well, you could do it with 5. Explain to me how you would do it with 5. Or explain any solution. So I want to code as input a vector that I derive from 5 consecutive residues. Can anybody propose a way of doing that? Yeah?
I mean, you have 24 amino acids.
20 amino acids, yeah.
And you have, if you use 5, then you have 20 multiplied, 20 powers 5.
So we have 100 input units. And every input unit would then be for one of the amino acids at position 1, 2, 3, 4, 5. And essentially, of all the vector I put in, I would essentially have 5 units that have a 1, because 5 amino acids are there for this particular case. And I would have 95 that are 0. Okay? Because every single position codes for 20 amino acids. And if it's an alanine, then the alanine unit is on. All the other 19 are 0. Right? Okay, that is one way of doing it. Now there's a tiny problem with that way. The tiny problem with that way is, say my central, typically what I do in this window business, I ask, what is the secondary structure of the central unit? Okay? Now, how do I treat the first residue? So, if in my window of 5, the first I can easily treat is residue 3. But how do I account for residue 1 and 2 in the beginning of the protein? Any idea? Simplest idea would be to ignore them. And some people have done that. But let's just say this is not an option. How could you do it? Anybody? Nobody has an idea? So again, the question is, how do I code for the fact that there is something before the beginning, somehow, in the protein? So residue number 1 in a protein, the first residue in a protein, should become the central residue in a window. I have a problem to find my central finger here. So the first residue should be this, meaning these two here, they do not correspond to anything in the protein. What could I do? There are many things I can do. Any idea? Insert a loop. Yes, so that's what I said. That is okay. That's the point taken. So rather than ignoring them, you could say I predict the most frequent state at that position, and it turns out that in fact the probability of loop at the first and last residue, I mean it's the same thing, it's symmetric. So the first residue and the last is the same. So predicting loop would in fact be more often right than wrong. So that's a clever idea, and people have done that. But let's go back to something that is more formal. That is, hacks are good, but at least I want to put it somehow into my device. Any idea? Put the fact that something is before the residue. So I have 20 units that code for 20 amino acids. How could I code for no amino acid, or for the beginning of a protein, or the end of a protein? And those two are equal, because there's some symmetry involved here. Any idea? No. No? Is it really so complicated? Or is it so simple? Can somebody give me, is it simple? There's no no. So, is it complicated? There's no yes. Is it too early in the morning? Do you want coffee? Or tea? Or a splash of... There's a lake. Okay, there's presence in the room. Well, one way of doing it is I said, when you have an amino acid, you have one of the 20 units on and 19 off. Well, one way of doing it is you put them all off. The problem, the way the neural network works, you multiply something with a 0, and that's not giving a lot of contribution. So this is not really, the network cannot really learn that. It can learn the absence of a signal, but it's a little bit more tricky. It could work, but it's not clear. Alternatively, what you could do is put them all on. But that would be an extreme signal. That may be sort of oversteering in some sense. And another one is you put a 21st unit. So the 21st unit, if a protein is 100 residues long, and you have a window of 5, you add 4 in the beginning and the end, you add essentially 4 units that are used only for that purpose. That may or may not work, but at least it's sort of an equal treatment. Okay, so we have 21 times 5, if we had a window of 5, we have 21 times 5. How else could I code for the amino acids? And in fact, that is essentially what you see here. there is the vector of 21, so this is the additional unit here, the spacer unit. In this particular case it's not 5, it's 13. In brackets you see the secondary structure that is to be learned from the supervised training. In this particular case the central residue is a proline. That in fact happens to be in an extended state, so in a strand. And the system is supposed to learn that if that is in the center of this unit, then this unit should go on. Okay? Now, do you have any other idea how you could code for the 20 amino acids? So this is one way of doing it. And in fact, it's the way that is most efficient. But do you have any other idea how you could do it? How would you do it if I told you that we don't have enough data points to train this device? Well, the first thing then you would do is you would shrink the window. Rather than 13 as on the whiteboard, you would use 5 or something like that. But let me say that the observation is 5 is not enough. You really need 13. And still you do not have enough data, because for every single input unit you have a connection to the hidden layer. And the number of free units is input times hidden layer. That gives you somehow, since that on this layer they also play a role, but the number is much smaller here because the output is much smaller than the input. So ultimately the most important issue here about how many free parameters you have is times hidden layers times the input. So if I could reduce the 21 presentation substantially, I may gain something. How could you reduce it?
You can group them up, for example, by, I don't know, substitute.
That's a great idea. So essentially we're looking at biophysical features. We say, for instance, I'm not going to code the 20 amino acids. I'm going to classify them into hydrophobic, polar, charged. And then I essentially have 3 units. 3 plus maybe the spacer. That is one way of doing it. Or I could even say that you can be hydrophobic and polar, or you can have several biophysical features. So I replace it by maybe 10 different biophysical features. And to me, that looked like a very promising approach. It did not work. My impression is it did not work because you introduce implicitly correlations between the vectors. So every single amino acid essentially is a part of a vector. And when you look at 2 13-mers in the protein, you have somehow a similarity between them. And by putting the biophysical features into these vectors, you introduce a different activity, as you may call it. So the number of ones that is on, for instance, one example is the number of ones that in this biophysical coding that you have for every single residue may differ. So if you allow the amino acid to have different biophysical features. And that creates correlations that is difficult for the network to learn. That's my impression. Anyway, let's get back to this simple solution here. And let's just see what it does in terms of performance. The performance of this neural network is essentially similar to the statistics, 62%. So there is no big gain. When you believe that just doing machine learning and you do it better, then that in fact is wrong. Now, there's another aspect, and I'm afraid that somehow I made a mistake in my slides here. I don't know why. When you look, remember the other issue was that beta strand was predicted at levels almost of random. And when you look at the fraction of correctly predicted residues that are strand, so I'm going to show you a pi, and due to mistakes in my slide here, the pi is not visible, but on the next slide, what you would have seen if this slide would be right here, is a pi here that shows you that of all the correctly predicted residues, most of them were other, then the second most often was helix, and very few of them are strands. So strand again is predicted at levels almost random, which is completely compatible with this statement that strand is less local. Now, when you look at the training dynamics of the neural network, then this is what you see. Oops. Sorry, there's something wrong with my slides. What you see on this axis here is the training time. At step of 1, I present all the samples that I have once. Here is the performance. The performance is separated out for helices in red, other in green, and strand in blue. Can I have a few observations what you see? Again, this is training time, training performance for the 3 secondary structure states. When you write a paper, a manuscript, the results section is about describing what you see. Say this would be the figure that you want to present. Then the first task that you have is to state what you see. Can you please state what you see? Can people, yeah?
After a short time, the results remain the same.
So we have somehow the situation that, again, what I have to observe here, we have already 20,000 steps done here. But there is some sort of convergence here. Is one observation. And that is the first important observation. Any other observation? I'd like others. So I'd like everybody to sort of have a little bit of an observation. Not everybody, but several people. Yes, that is correct. Yeah?
After 2 training steps, the strand is, the helix drops and the strand increases.
Yes, so strand increases very much here, while helix drops. Okay, next observation. That's an important one, because it somehow sets you off to expecting something in the next step. So if you now took this observation to expect without seeing the rest, if you only saw the first 2, you may expect that you somehow exchange strand for helix. So then you would expect that this goes down. That's not the case. Or that this goes down while this goes up. But this is clearly not the case, right? The next observation would be helix goes back up. And strand continues to rise. In fact, if there is sort of a swap somehow, then it's between green and red. So there's some alternation between green and red. It appears as if when red is high, green is low, but as you correctly observed, essentially between red and green, you reach somehow a steady line, and you may even sort of visually, it looks as if the average between red and green is constant after the first step, so it's up from step 2 on. Actually, even after the first step. But you still see something in strand. Strand is improving. So the improvement of strand, that's the next thing you observe, is not on behalf of green and red, of other and helix. Strand just keeps growing. And they keep doing their alternating business there, their alternating dance. Maybe there is some saturation here. Maybe here, maybe here. Not so clear, but at least for a significant amount of time, strand still increases. Why could that be the case? Nobody? Why could that thing not learn strand? Machine learning has a problem, or this machine learning device has a problem to learn something. Why could that be the case?
For that same region where strand is increasing somewhat, other is decreasing, so maybe some of the other.
Yes, so that is the point. So somehow where this one increases, and that was the observation here in the beginning, the others decrease, and maybe it's a payoff event. But overall, the payoff event seems to be, if you go look further on, the payoff event somehow seems to be between red and green. Like strand, at least to some point, at some point here, at this point or that point, it depends a little bit where you want to put yourself, but at least beyond point 2, strand continues to increase, and not on the shoulder of green and red. Why could it be that strand? is a problem here. Anything, I told you this morning, something comes to your mind. Any suspicion? You can also give an argument and shoot it down, because the argument that may come to your mind the fastest could be shot down, yes?
Probably that's a matter of local information.
Exactly. Maybe that is exactly, I'm sorry to misuse you, but I hope it makes the lecture more vivid. But this is what comes to your mind immediately. This is because it just confirms what we said all the time. Strand is not local. So that is why it's much tougher to learn. But there's a clash. On this slide you see something that clashes with that interpretation. It's exactly the interpretation that anybody who's in the field had when they saw this slide for the first time. But there's a problem with that assumption. What is the problem with that assumption? And again, this is once you have the answer, it's very obvious. But science is very often about stating what you see to yourself and to the reader. Because discovery is to find the mistake in the obvious, or to find that the obvious is true and nobody ever expected it. We are happy to see this curve and say Strand is more difficult, hence it's less local, hence we see what we see and move on. But there's a clash. There's a problem with that interpretation. And many people have overlooked that, although it's totally simple. So once I give you the explanation, you will say, of course. But it's very simple to see. Can you see it?
Doesn't it give one in total?
Sorry?
Doesn't it give one in total?
It doesn't give one in total. That is right. So but this is not the point I want to make. So again, what I show here is the percentage correctly predicted. So they don't add up to 1. Not all residues are correctly predicted. What I show is the fraction of all the residues in helices correctly predicted, 80% here. The fraction of all the residues in other state correctly predicted, 70%. The fraction of all the residues in strand correctly predicted, 10%. Again, random is 30 or something, or 35 or whatever you put it, but this is much, much higher than that. So why what is on this slide here that puts a question mark to this simple and common explanation that is simply because Strand is less local and more difficult to learn? Is there anything on this slide that sort of puts a question mark on that statement? There's a lot for that statement. Helices and Strand are, helices and other are, yeah?
In most cases, the range should go from high to low.
Well, these here. I'm talking about the blue.
Yes. As I know, the graph that shows the neural network learning in most cases go from up to down.
Oh, so then they show error. So what you talk about is you simply saw error graphs. So this is accuracy. I call it performance. And I define what it actually is. Yes, typically you see these, this is correct observation. Most people would plot it the other way around, 1 minus. But again, I'm sorry. What is the issue going away from this slide and saying this proves that Strand cannot be learned? It's so obvious, nobody sees it. Yeah? Yes?
Do you mean that since it's difficult to learn Strand, it takes lots of training samples, like more number of generations to learn, and that's a lot of overfits in data, and that's the performance in other like algorithms and anything?
This is an interesting one, because you essentially say everything, but you don't say it explicitly. So the answer is in exactly what you say. But I can also say, if that would be an exam, I could also find reasons why I could say you didn't say it. So what she said, for those of you who couldn't hear her, is that in fact it takes much longer to learn the Strand. And she also said something, and I'm not entirely sure whether you're aware of that. You said this is because it's less often, less frequent. You said something like that. The main part of that statement is, there was somebody else who wanted to say something.
I don't get the question because the performance of predicting a strand is a lot lower, like in general, the threshold is a lot lower than predicting a helix or another form.
Yeah.
Does that imply that?
Yes, so that's an interesting observation, and it's certainly a limit in what I'm trying to establish here. He says that he doesn't get my point because after all blue is lower than red and green. Correct. After all, I said blue would be less than 40%, in this particular case a little bit higher. But the main point I want to make here is a slightly different one. And you're right, and I now see how that shields the message. The message that I wanted to give you is, yes, Strand is really local, and yes, it's not local, and yes, it's complicated to learn it. But it does increase. Okay, in this particular case, it goes over 40%. In fact, this would already be the best Strand predictor in the world had it been published. It still is true that blue is below, but she also already said something else. Again, that is the slide that I didn't show you before because of problems with the slide. This is the correctly predicted helix residue. Of all the correctly predicted residues, 62% of all the residues are correctly predicted. They make the pie here. White are other, blue Strand, and helices are red. When you look at the distribution of the same in the database, this is not correctly predicted on this side here. This is simply how many helix residues do you have for training, how many Strand residues. They look surprisingly similar. You said that maybe this is because there are fewer Strand residues, and this is when you look at these pies, this comes exactly to your mind, right? So what we'd like to do is to have this distribution. We'd like to predict something like this. How could we do this? Well, one way of doing that, when I train a neural network at every single time step. Is there a question? Yeah?
If you want to predict like the normal distribution of those three.
Why do I want that? Well, ideally, let's just see whether I can. Okay? Let's put it like that. So my first observation is that my prediction accuracy resembles the distribution. And I actually want a better Strand prediction. And one way to think about a better Strand prediction is by saying, well, I want them equally predicted. Okay? Let's get back. That's a very valid question. I'll get back to that in a minute. So how do I do that? Now again, at every single time step in a normal neural network, I present one sample. And rather than doing that at random, one sample, at every single time step choose one residue from helix, one residue from Strand, and one residue from other. And I simply sum over the differences. Okay? So effectively that means that I oversample Strand. Because I sampled Strand at 33% of my samples, and obviously observed is 20%, I oversample it. And I undersample the other class. Now, when I do this, and there's something totally wrong with my slides. Or is it my machine? So when I do this, that is what I call unbalanced. So it is in the number, it's in the database, and this is the balanced training. Okay? Now, there is no difference. The gentleman who observed that the blue is lower, on this side, the blue is no longer longer. They are equal. In fact, over the entire range, there's a very smooth behavior here. When I look at the pie charts, it's totally equal. 1/3 of all the residues correctly predicted are strand. And we get back to your question, why do I want this? In fact, you get back to that looking at these two numbers here. I get an equal prediction of all three classes, but at the expense of the overall accuracy. The overall accuracy drops because most residues are other state. And since the other state is overrepresented and predicted better here, I get a higher number. But I created something else. I created a neural network that predicts strand as accurately as helix. Yes, in biophysics, it is clear that strand formation is not local. But I'm not using any global interaction. All I do is artificially, in some sense, in my computer, change the sampling. And I create something that completely appears to contradict the biophysics. It's not really contradicting the biophysics, but it shows you that the reason why strand is predicted at a low level of accuracy has nothing to do with the biophysical explanation. Because I can immediately develop a method, I can predict them at the same accuracy. Whether I want that is a different story. Because somehow what I want is sort of compromise between these two extremes. So I do want strand better than random, but I also want a higher number. And somehow I will try to find a compromise between these two. But the major issue here is that if you understand your machine learning device and you observe it while it learns a problem, you can learn a lot about the problem. Prediction is the acid test for understanding. Whether you understand or not is only answered if you can really predict, and by looking at the prediction device you can learn a lot about a problem. And the statement that strand is predicted less accurately, or just less than 40%, just about random because it's not local, it's true it's not local, but not all sound explanations are right. Now, this is an important point. It's an example where you can use a prediction method to completely prove that some explanation that makes sense is not right. Let's get back to the next issue, the issue of short segments. So I said that the problem of a prediction is, in this case here, when the segments are too short, you don't know, is that a long helix, is that a long strand, do I have to cut this E here? On average, the predicted strand is 3, the predicted helix of the neural network, the predicted helix is 4, and those are the observed averages. So the helix is twice as long. Now, any idea how that could have happened? Why is it that these segments are predicted so short? Why is it that the system cannot learn the length of a helix? Any idea? Any suggestion? Any opinion? Any suspicion? Okay, let me repeat. What I do have to do is, this is sort of the connections here, the error, and that's the way you wanted to see it, so the error goes down. We lost him at this point. So now, in order to not get trapped in local minima, and not to avoid all kinds of other issues, ultimately you can visualize them as local minima, what you have to do is you sample at random from different places in your set of training. So you have, say, 20,000 residues in your training set from 100 proteins, and you sample from protein 1 at position 15, and the next one is from protein 42 at position 63 or something like that. Any idea now why segments could be predicted too short? Your device never sees correlations of adjacent residues. At every single window that it sees, it predicts the secondary structure for the central residue. There's no feedback loop that says, okay, if you have a single helix residue predicted, that cannot happen. Helix has to be hydrogen bond formation, so I plus 4, it has to have at least 4 residues. This system never sees that. What you could do is you could sample in training an entire helix. The problem is this brings you in the training dynamics too close to local minima, and it's not easy to get out of it. It's not what you want to do. It creates new problems. Although we have a sort of uphill move to step out of local minima, this still is a bad idea. Now what else could we do? So how could we somehow put those realities that a helix has to have a minimal length, and that even strands are typically not formed between, sheets are not formed between 2 residues, but they are sort of extended. Any idea how we could put that into the system? Yeah?
Maybe take more hidden layers so you can memorize the last prediction.
Yes, so that in principle is a good idea. Now, the idea here, he said 2 things. And one I believe is a good idea, and one is not. Well, actually, you could also make that work today. So, yes, we can do that today. In the olden days, you could. So, essentially, he said 2 things. 1, put the output back in. 2, put it back into the hidden layer. Putting the output into the hidden layer is not that easy to do in terms of the way you compute the error and the way you change the connections. It's much easier to put it back into the input. Then, because for the input you can somehow trace the error, the dependence of the input on the output, and that is the dependence that you want to influence. And another way of doing it is much simpler. It's a hack solution if you want. In this much simpler solution, you have here the neural network that predicts the central residue V, the neural network that predicts the central residue P, and the neural network that predicts the central residue E. You take the output, you add a spacer unit again because you have a residue before the first. Put those, essentially in this particular case, 3 units together and put it into another level, into another layer of network. So this layer of networks here would simply predict sequence to secondary structure. And the second layer, or the second network here, would put predicted secondary structure to secondary structure, predicted secondary structure. And that would essentially learn that something like EHE cannot be. So you cannot have an isolated H. Indeed, simply doing this, and you train with the same data set, you train this network here and this network with the same data, just repeat. And this is a much simpler network because it has fewer input units. So this one has only 13 times 4. And what you get out is this. So this is the first level, and that's the second level. So that is much closer to the observed. And when you look at the overall length distribution, you will also in detail see that the secondary structure elements are much closer to reality. It was, this idea of a second level network was invented by Shannon-Sainovsky, but it was invented for a different purpose. It was invented to increase performance. It doesn't increase performance, but it does exactly help to predict better length. And Shannon-Sainovsky had overlooked that simply because that is what they didn't look at. And they had invented it, but had not really used it because the overall accuracy didn't go up. So there was no point for them in doing it. So the lesson from this one here is you sometimes have to really look into more detail and into different scores. The overall accuracy may not be the only aspect of performance to monitor. In fact, there are often different scores. And typically the challenge for your thesis is you create a lot of data. Find the few that you put into the manuscript. Focus. And find the ones that you focus on also when you sort of direct what step to do next, that's the challenge. And the only way to do that challenge is give yourself time points at the end of the week or whatever it is, and report to yourself, if the supervisor doesn't force you to do that, to report to yourself, what is it that you actually found out in that week? What is it that if you wanted to report the one thing, what is that thing? And sort of come up with statements, descriptions of the results, statements, and see how much is needed to in fact verify them or support them. Okay, there are other ways of doing this. And clearly the most advanced way of doing this comes from Gianluca Pilastri. It goes back to Paolo Frasconi's idea that you, now in this particular case here, the input is here, the output is here, and you essentially feed the input back into the output back into the input. That essentially is your idea. And Paolo Frasconi and Pierre Bailly worked out the math of doing that. Okay, all of that solves the problem of beta strand, it solves the problem of length, of segments, too short segments. But it's still only doing 60 plus something accuracy. So the overall problem, we have solved two problems with this. We have not solved them with a machine learning device, but we have solved them because we understood the machine learning device, and the machine learning device was a very simple mechanism to eradicate misarguments that had been there for decades. The machine learning device helped find mistakes. It was very easy. You just have to understand your machine learning device. So you have to somehow get more data into the input. How can you get more data into the input? Making the input, yeah? Question or statement?
I don't know if it's related, but I still don't get how we use this, how we select the window. Is it related to the question?
No, that I believe is not related to the question. Let me get back to, thank you for asking it. That is as good as any other. So essentially you're asking, how do I get this input? So what we want to do, and maybe I do get back to the first neural network that I showed you on this. And it's essentially, I'm showing you on a neural network is the same for any other device, including statistics. This, in some sense, is the answer. So ultimately what you want to do is you want to look at secondary structure for a particular residue. And you want to see how its local environment influences that secondary structure. The choice of doing that here is to put them as input into a machine learning device. Now the way you do it in a machine learning device, say the sequence would be dr, q, and blah, blah, blah. So that's 13 consecutive residues. And say that's the first here. Then residue number 7, in this particular case, is a proline, a P. And you would predict or you would learn the secondary structure for residue 7. Now, in order to predict secondary structure for residue 8, you move one further. Simply take 13 consecutive residues, one step next. So this one goes from residue 1 to 13. The next would go from 2 to 14, 3 to 15. So you move this window, you slide the window through the entire sequence. Okay? In order to do this, you will have this issue, how do you treat the D as the first residue? And that brings us back to the spacer issue. Because if D moves to the center of this window, the 6 residues before the D are no amino acids, because there is no protein before. So for those, the spacer unit here is going to be on. And again, if anybody doesn't get this, please ask again. This is very important. This is again the kind of thing that is asked in many exams. And again, it's again and again misunderstood. I'm sorry that I did not take enough time. Thanks for asking that. So again, if you wanted to predict the secondary structure for the first residue, you have, in this particular example of 13 consecutive residues, you have 6. So before, whatever is before 6 here, you have 6 spacer, non-amino acid, non-protein units, which here in this particular cut out are these. And then you slide the window through. That means for every single protein of n residues, you have exactly n input windows. Because every single residue, every single position in that protein will be used once in the center of the window. Is that clear? Anybody unclear about this? Yeah?
I'm sorry, I was a little late this morning. I'm not clear about the relationship between the different colors and...
You mean these colors?
Yeah.
So this is the output layer. In the output layer you have helix, strand, other. This is the input layer in yellow, where essentially you put the sequence in, because you want to predict secondary structure from sequence. In a sequence, out of secondary structure, you learn by, in fact, adjusting these weights such that the output you want is the output you get for these samples that you know for your training set. The hidden layer is called hidden because it has nothing to do with the real world. World is just a computation. In is visible, out is visible, in between you do some computation, you have the free parameters in between. What you optimize are the green lines. What you can change, you cannot change the input, you cannot change the output. I mean, you have to adjust the output such that it is what you want it to be, what you know it to be. You supervised training is supervised learning. That means for 20,000, in this particular example here, I showed you the training dynamics for 20,000 residues. For 20,000 residues, you know exactly what you want to get out. And that is the score I showed there. 80% of the helical residues were correctly predicted. Is that answering your question?
Yes.
I do not care why you don't get the point of the way this works at this point. We were too late, you slept, I really don't care. But if anybody is not clear about that, please ask now. Yes?
So could you explain it one more time?
Yes, sure I can. Again, as I said, I really don't care. But this has to be clear for you. So this is the input. The input, you want to map sequence to secondary structure. Secondary structure you want to predict in 3 states. Helix, strand, other. That's why you have 3 output units. Input is sequence, as I said, and we're going to take, in this particular case, I show you it's 5. Here on the whiteboard I have 30 consecutive residues. And I want to predict the secondary structure state of the central residue, which happens to be a P in this particular case. Okay? So in this particular case, I know from my example, this supervised learning, I know that this proline is at an E. So this network has to learn to predict this P. When it gets this input vector, this unit should be on. If I randomly choose my connections, this most likely is not the case. Randomly is 33 or 35% of the cases, so in most cases, 35 is less than 50, in most cases, this will not be the top unit. So the only way I can make it become the top unit is I adjust these connections. And I adjust these connections by simply going down in a gradient descent, or in a derivative of the error with respect to the connections. I showed you the formula for that last Thursday for training, and I'm going to show it in a few slides again. But that's not the important thing. The important thing now is how do you code for the input. Now, again, we now see the larger yellow ovals here. Each of these is for one residue, one position in the protein. And again, the central one here is for the proline. Now, each of these ovals consists of many input units, because each of these codes for one position in the protein. Protein position 1, 2, 3, in this particular case 7 is the proline, just made up. I mean, this is a real example, but the position here is not. And since we have 20 amino acids, the simplest way to code them as a vector is by using 20 units. The 20 units being such that if that 19 are 0, that's the yellow supposed to mean here, and 1 is black, the P. This is the central P, 19 zeros, 1, 1, the P, because there's a proline at that position. There is another unit here, I call it the spacer, that you need, because if that really were residue 1 to 13 in my protein, everything before the proline could not be in the center. How do I put the residues V, F, G, Q, R, D into the center? I have to move it, but then I have to say I moved by 1, so the V would move to the center here. Then there would be 1 empty, because that is the first residue. Residue minus 1, then you move 1, be the 0th residue, so to speak. And that's what I call the spacer. So when we move D to become the center residue, then 6 residues above it are spacer units, so it's not protein. And the same will be true if the last unit in a protein is the center. Then 6 after it will be the spacers. Is that clear? Again, anybody who has, I say it again. Well, one more time. Yeah.
I got questions about the input. So what do you mean by a single yellow dot? Is it a single amino acid or several ones or a sequence of them?
So this particular case, again, so imagine that when you write down the sequence of this protein, the sequence reads D, R, Q, G, F, V, P, A, Y, V, K, K, L, P, R, K, T, or something. Okay? And now, here I have 13 consecutive. Let's just play it on 5. My first window that I can create that has the D in the center, or let's play it on 3, would have 1 spacer, the D in the center, and the R. Just that sequence. And then I move this by 1 over. My second window would be the D, R, Q. My next one would be R, Q, G. In the center would be, in this particular case, the R, in this particular case, the Q. And when I say R, it is in the vector of 20, this unit would be on. All others would be 0. So, meaning that for 13 consecutive positions, or in this case here for 3 consecutive positions, I would, of the entire possible input, 13 times 21, I would have exactly 13, or 3 in this case here, that are 1. All others would be 0. For every given sequence. Is that right? Clear? I'm not going to say the same thing again, but if you make me say it in a different way, I'm ready. Yeah?
What does the parenthesis mean? So D, parenthesis L, what's that?
Oh, so again, in the parenthesis what I show here is the actual secondary structure that this particular residue has been observed in. So this is my supervised training, and in this particular case, this is an unusual case, but it actually is really true. Usually you do not have an E going into an H. In this particular case, you actually had it. So this part is a cutout from a real existing protein and a real sequence. In fact, it's one of the ones that I showed many examples here with a 310 helix. You may know. I don't show it here. On one of my slides it is. But so in this particular case, what I have to learn, that's why I say I want the E unit to be on for that proline. Because that proline is in the E state. Okay? My second level would essentially put this output as input. Okay? Whoop, 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 whoop. Here it is. On the second level, whatever I predict from the first level, so this is a complete neural network as I described it, that predicts each HEL as output. And I'm now going to use that as input to the second level to learn correlation that's of the type HLH cannot be, or HLH can be, LHL cannot be. The helix has to be 4 residues long, and one way, so I show only 3 here because I did not have enough space. I will actually play that on more than 3, so that I can sort of see an entire helix in there. And again, I need a spacer unit, that's why the input here per residue is not 3, predicted secondary structure, but 3 plus spacer. Both are clear? There were questions in the room, yes?
So do we know how this hidden part is working, or we don't know the blue one?
So, it's not shown here. This is the training dynamics. But, so essentially what you do in the hidden part, so if that here is my yellow, and then my hidden part here, so we have an entirely connected network for simplicity, and then in this particular case here my output, and so forth. So what you do here is you have an input unit I, or let's call it J. You multiply it with the connection J, you sum over the J. So you sum the value from here times this. That's this input sum here. Oh, I need to move. No space. So far what we have here is let's call it K. K on level 1. So level 1 is these connections here. I sum over the K, and I will do a sigmoid function. That is, for instance, a hyperbolical tangents. It's the simplest way of f equals hyperbolic of tangents. So this is essentially the output from one of these units. Now, I have many here, so I need a J. So that's the output from here. I multiply that with a second level. So now we have a J here, and we have an I, and we sum over the J. And we do the function again. That's the simple math. So the hidden layer is doing exactly that. What you see on the whiteboard up there is the way you decide in the next step how to reduce the error. You simply gradient go down such that the error decreases. And the error, ultimately the error function is missing on this slide here. The error function is essentially the output that you have for a given sample minus the one you want squared. Yeah?
So we have K blue bubbles for the...
yes.
And what was the J?
No, not K. So, but you're right. K is the index. So I make it a capital K, and then you're right.
And what is then J and I?
So we have I would be for output unit 1, 2, 3. The J would simply sum, because these units are all completely symmetric. So the computation is done essentially not quite symmetric, because this one sums over anything that has been coming to it from before. But this is simply a propagation of the input. It's totally simple. So in that sense, it's also symmetric. But for every single unit, they do two things. They sum over all the input that they get, and then they apply the tangents hyperbolicus, some switch function. In that sense, they're completely symmetric. So maybe we should not use the same symbol here. We call that an H for hidden units. So that would go over the hidden units, and this would go over the output units. Let's call it O. No, wait a minute. There's a mistake here. This is not right. I'm sorry, I gave you the wrong answer. This is not H. This is H. And this is N, number of input units. So this, in fact, the sum over the K is a sum in the example that I had over 13 times 21.
What was H again?
So H is the number of hidden units, the blue. So H is, okay, in the sense here I call it N hidden. And the example that I showed you is the 6 blue. In the network that I showed you before, I had 6 blue. Oh, where's it? Oh, good. Where was it? Anyway, you remember it. And this one, I took the same, so it's the 6. So the H here would be essentially the last step you do is you sum over all the connections. You pick any, this is I in my computation here. You pick a sum over all these 6. But again, so the details of how that is done are easily available on Wikipedia and actually given to you on last Thursday. Any other issue that is specific here to the input? While we are on it, the neural network essentially projects the input vector upon the vector of connections by introducing a hidden layer that allows to separate points by hyperplanes. That's simple. The same is true for support vector machines. You may have heard about SVMs. The computation, the idea behind it is exactly the same. You have an input vector, you have a hyperplane that distinguishes between the points. Can anybody tell me what the difference is between neural networks and support vector machines? The conceptual difference? Can anybody tell me anything where they're different? Well, the words. One is called support vector machine and one is called neural network. That's not what I mean. Anybody? Nobody ever heard about these things? Okay. So, a neural network. So I have 2 types of points here. A neural network would essentially find a line or a hyperplane. In this particular case, it's a 2D environment. Typically, it's not. The dimensionality is much, much higher. So it would find a solution that optimally separates out these points, and it would consider all the points for doing this. And this could be a line. There are many other lines that I could draw that would also separate out this. I mean, just to have a few others here, right? The support vector machine has the philosophy of saying, well, these points here are trivial. I don't care about them. What I really care about is to identify the points that are closest to each other. So you could argue, so these are the sort of points that are closest to each other. And now I design my solution such that it can separate exactly those points. Okay? What is the implication of that? There are 2 implications of that. One is good and one is bad. Can anybody sort of pick them up? Yeah?
It would provide a good result between these Burkhard regions, which are really close to each other, and but at the same time it completely forgets.
It completely forgets about all the other points. That is true, what you say, but in fact even there's a tendency to say it finds the line that is equally distant to, somehow finds the best point to separate these two. Now if I had this line from an SVM that only cares about these points, you say it would forget about the other points. But in this particular case it doesn't actually. You're right, and we may have overlooked some. Completely true. I may have thrown some away that were overlooked, but for this toy example that I draw on the whiteboard, I threw them away, but at no cost in some sense, because they are actually implicitly classified. No, not implicitly, explicitly classified. Because they just happen to fall on the extreme side. So it may work. You're right, this may be a danger, I may lose points, and they may be relevant. But as long as I really find the ones that are really closest, nothing on that line is likely to be avoided. In the future I may find points that are. But let's look at something else here. There are two implications that are immediate. One is I use fewer points, right? So I have always been talking about the number of free parameters has to somehow match to the number of points that you have. So if you're on a train of 20,000, you cannot have 20,000 free parameters. The number of free parameters should be substantially smaller than the training set. This would make it substantially smaller because you need much fewer. That's the upside. Can you imagine a downside? Just imagine that because of an experimental error, this actually was a cross. I thought it was an open circle, yeah?
So the disadvantage is that this method is not resilient?
It is?
It is or it isn't?
I didn't hear what you said.
My question is, so the disadvantage is that this method is not resilient enough, or?
You're too fast. Yes, I was about to say that. Yes. So this method, in some sense, you call it not resilient enough. The problem really is that if the two closest points may be closest, because they are identical, they should fall into the same class. You just made a label mistake. You made an experimental error. So if you make experimental errors, this method is ideal to try to mistakenly separate them, because it's explicitly finding the ones that are closest, which may exactly be the error points. So somehow the SVM has also a talent to zoom into your most problematic parts. And you could call that resilient in some sense to errors. Yes, that's the downside. So the upside is fewer samples. The downside, you need to be more accurate, or you need to rely more on, you can only, it will only work when you have fewer errors in your data. So for very noisy data sets, this is not the right solution. Yeah? So, okay. So I believe at this point everybody sort of understood the issue, and the question remains, how can I put more data into my input? So I correct on this issue of short segments, I correct on the issue of beta strands, but I still have only 60-something% accuracy. How could I put more input into more information into the input? I cannot make the window longer. Yeah?
The size of the window is always fixed?
So no, I could, so yes, sorry, let me repeat that point. I can make the window longer. It turns out what I add is more noise than signal. So yes, I can make it much longer, but it doesn't help me. So in that sense, I cannot make it longer. That is not the solution because it just doesn't work. No, the size is not. Oh, you mean for one particular choice? Yes, I'm sorry, I misunderstood your question. Yes, so I design a neural network for, in this particular case, the way I showed you, is 13 consecutive residues. Then that prediction method will always use 13 consecutive residues.
What if How variable?
We could take 15, we could take 20, we could,
Longer, but maybe sometimes use like 3, size of 3 and then size of 8.
So we could mix different neural networks and we could somehow average over those. Great idea, but didn't work. Tried that. So I also found that was a great idea, but it just didn't work. So trying to sort of sneak around the problem by allowing different networks to zoom into different aspects of locality in some sense. That I assume was your idea behind it, at least it was mine when I tried first, but it didn't work. So I need to find a way to get more information into a window. It does somehow work, but it does not do the major step. So we need, did that advance by itself or did I touch something? Okay, so the answer is on the whiteboard. But do you actually see it? Do you see what I mean by it? It's not obvious. So what it means, the title means that evolution has this information. What I show you is a slide, the HSP curve. I've shown you that one before. And this is again is something that I hope you will be able to somehow memorize, at least not exactly where things fall, but in terms of the general idea. Number of residues aligned, percentage sequence identity. You have 100 residues aligned, and if 33 of those are identical, essentially the two proteins have a similar structure. This effect of the blue you use for comparative modeling. And the thing that is important to remember is when you have more residues aligned, you need less sequence identity to make sure that they have similar structure. If you have very few, at some point you cannot distinguish. In the extreme sense here, for 11 consecutive residues, they are observed in different structural states. And that is back to some secondary structure is not determined only locally. 11 residues are not enough. 11 consecutive residues you could find in different structural environments, okay? And that's why it separates here. So for 11 residues, even identity is not enough. But again, so for 50, you need 50 or something like that, and you go further down. Now, let's pick this example here. I can change 67 of 100 residues without changing structure. I can also change only 5 and actually alter structure. So if I went to the lab and I changed 5 residues in a 100 residue protein, odds are pretty high that I mess up the structure. So how come that I change 5 and I mess it up, and nature changes 67 and maintains structure? What's the answer to that question? How come nature can do something that I cannot do? Well, nature is smarter. But what is the constraint, or why is that the case? There's something I don't... that was not an answer. Any idea? Well, the point is that sequence changes happen through radiation, through copy mistakes, from parents to their children, copying the genome between father and mother. Mistakes are introduced. So sequence variation is something that happens. Now, in fact, sequence variation is something that is the base of evolution also. But if we mess up, then that protein has no future. And sometimes the carrier has no future, in extreme cases is not born. So, put differently, there is a high incentive for neutral changes. There is no pressure to make a neutral change over an effect change. But the effect changes, many of them will not survive. We will not see them. So what we see is a subset that is completely not random. What we see is a subset of the unlikely event of neutral variations. Only those survive, or they have a higher odds of survival on the level of a protein. Survival in the sense of being observed, right? That means nature is much better than I, because it's optimized neutral changes. Nevertheless, for one particular protein of 100 residues, which are the 67? I cannot randomly pick them. Nature optimized which ones we can change. Which residue can I change in what position against which other amino acid? That is the information that is somewhere in this profile. That's somewhere in this evolutionary exchange matrix. That is exactly what nature, when I compare a family of proteins, the information about what is relevant where is in there. I showed you this example here. We have an alignment of a protein family, and in this alignment there are sort of 1, 2, 3, 4, I'm sorry for the purple is no longer clearly visible, 4 relatively conserved columns. They matter more for that sequence, and this is the way I introduced the idea of a profile. And in this particular case here, when I look at the 3-dimensional structure, and those are 2 different sides of the same molecule, the first of these conserved columns is this residue. The last of these conserved columns is that one. You see the last and the first, they are close in 3D structure. When you look at 5 consecutive residues in the back here, what you can observe in these 5, or what you do observe in these 5, in terms of the exchange in that family, has to do with what happens here. Because they evolve as a 3-dimensional object. They don't evolve as sequences. Proteins function as 3-dimensional objects. So their reality is always this bond, is always this contact. Their reality is always that this one here is next to them. They see proximity that on the sequence you don't see. On the sequence you don't see that this one is in contact with that one. But because whatever you see in this local environment, whatever is around it, has to do with what is here, because they are close in 3D, means that the observation in this profile is impacted by what is here. Okay? That means that by just looking at the 5 consecutive residue profile, the evolutionary exchange, whatever I see in this 5 here, tells me something about somewhere else in the protein. This is like the raindrop in which you see the world. Okay? There's implicitly there. You may not be able to see it, but implicitly it's there. And implicitly it means you sort of transfer the problem that is like chaos, that is difficult to solve, to the kind of problem that you can solve by, if you want, averaging over different species. By looking at what was possible in evolution. And that means that this evolutionary profile is exactly containing the information density that we want. So all we need now to do is, instead of put amino acid with 1, 0, and 19, or plus spacers, so 20 zeros and 1, 1, we now at every single position, 1, 2, 3, in this particular case is the same sequence that I showed you before, with a proline in the center. I simply, in an alignment in the family of proteins, so this is protein 1, 2, 3, 4 that are similar to this one here. Previously I used only this sequence, and now what I'm going to put in is just how, for every single position, how often do I see a D? So this particular case would still be the same as before. Here it would be the same as before. But here I would have 20% E. I observe an E here, but ultimately the input would only be 20% E and 60% A. Okay? At that point. Is that idea clear, or is that solution clear? How I use the evolutionary profile here? Is the slide clear to you again?
How do we get the alignment?
Okay, so this, I did not tell you, this is an open question, but the way we do the alignment is exactly like the methods that I told you before. So you do pairwise alignments, so you take a protein, in this particular case you start with this protein here, you run against the database, you find all the pairwise alignments, then you build a profile, like this one, you run again, that's the PsyBlast for instance, that does it, sequence, then sequence profile alignment, and then ultimately you decide whatever is above this threshold, the blue in the curve that I showed you before, sequence identity versus, alignment length versus sequence identity, and whatever was blue in this curve before, where I said 100 residues, 67 can be changed. In that curve, bless you, whatever is above that curve I take and I sum. Clear? Okay, so here's the process described in a little bit more detail, but it's essentially exactly what I said. There are some additional problems. There is this issue that there was a balance that had 60%, the unbalanced that had 62, so I can just take a bunch of neural networks and average over them. In fact, if I have different classifications, and I average over different classifications, as long as there is some white noise, I'm going to improve. Is that statement clear to you? Do you have an idea what white noise means? So white noise, for those of you who do not, I'm not entirely clear. I hear several yes, I hear no. I move on. I don't hear yes. Darn, this is complicated, guys. I need steering. Okay, so one way of putting white noise essentially is non-systematic errors. And the part that is non-systematic, so for instance, you always make a mistake on a strand because the way you measure. That would be systematic error, and that you cannot fix. But if it's just a random mistake, then the more you average over devices that are not correlated, the more you will improve. You may not improve by much, but averaging always improves just a little. Ultimately, the payoff may be that you have to have so many devices that you use so much CPU that it doesn't pay off. You have a tiny gain and a lot of CPU. But in principle, this rule still holds. Okay, then I do this for two different levels, and in this particular device is sort of the modern way. There's something else added to it. Let's not get into that, and let's just see how well we do. And these examples are from essentially the device I showed you before. So that's the observed secondary structure. The white spaces here are other, the state other, for simplicity not shown. That's Schau-Fassmann, first generation, that simply looks at probability for a single residue. Gore looks at a probability, is in fact Gore 3 here, technically, probability of the same but for windows. And you see that this is 59 for this particular case, which is above the expected average of 50 something. This is 65, which is above the expected average of 60. And the neural network that uses evolutionary information is here, and it's 72. How do I know whether 72 is the average? Well, I look at the distribution. In this particular case, the distribution is this. So I have a bunch of proteins, and I cannot remember how many those were, I believe 500, ballpark 500, and the average is roughly 72. Okay? So now, what you observe here is that for some proteins you predict very well, right? And for others, you predict at levels that are, so 30 something is 35 is random. So there are at least 3 proteins here for which you predict at levels below random. That's really bad. So overall we get a method that the first observation is that the average really is 72, and that the average really is much higher. So the argument that secondary structure cannot be predicted better than 60 something percent is wrong. You can. And what we put in is still only a window, right? Okay, the window knows something from the rest of the world. It uses evolutionary information. But we can get higher. How could we possibly identify cases where the prediction is very, very, very poor? So most extremely here, I have a case that is way worse than random. So for that one, I would do better just throwing a coin. Why not do that? Why don't I just take that protein and throw a coin instead? Yeah?
Maybe when the proteins differs a lot from the profile you use for training.
So you actually have a couple of issues in there. So first of all, what I hear you say, so let me repeat for those of you who didn't hear it. Maybe for those there was a problem in the profile, or it differs very much from the profile. So the first thing I hear from you is to look at something that is going on in my protein and find reasons why that goes so wrong. And that is exactly what you ought to do. This is, I'm showing you an example here about secondary structure prediction. For whatever you do, try to look at distributions, try to look whatever you produce at outliers. Very often, outliers can teach you about bugs in your program, okay? If they don't teach you about your bugs in your program, they teach you something about limits of the method, or may teach you. Maybe you can learn something about your method that you can improve. So look at what is going wrong here. Can you see anything that is wrong? Maybe, in fact, this is a case where I don't have a profile. This is a case where I don't find in the alignment similar ones. If that would be your finding, what would you do next? If you would find that indeed for this one here, in the alignment you find nothing. For this one, in the alignment you find nothing. Then you would suspect that the problem really is that when there's nothing in the alignment, the method that is trained on the alignment performs very poorly. How can you test that? Okay. Yeah? Yes, you can. Well, if you look at the sequence profile alignment, so essentially what you do here is a sequence profile alignment, and your suspicion was that maybe, or this is I'm sort of twisting what you said. It's not exactly what you said, but I'm twisting it. That could be one possible outcome, and for two of those, you in fact find yes, it's true. My alignment goes wrong for those, but how could you find out whether that in fact is the reason why those are so low? Nobody? Again, I'm talking about secondary structure prediction, I'm talking about computational biology, but the idea, the answer is based on an idea that is true for any kind of thing that you do, not only machine learning. If you assume that the problem is the alignment, then you throw the alignment away for everything. The simplest way, if you come up with an idea what could be the reason, you have to test it. This is science. And testing it means, well, you're going to try for everything else. How if I don't use an alignment in any of those? If that would be the explanation, then the whole distribution should be moving very much in that direction. Indeed, what you observe is that the distribution does move. But in fact, it moves by less than 5 percentage points. And you clearly see that this is not enough, but would then see that this is not enough to explain this outlier. Okay? That's an important issue. But my question remains. But he said something else, and I'm going to answer that question too now. So you cannot just pick this because you don't know for the next prediction case the answer. You're applying your method for a case for which you do not know the answer. So you have to find an intrinsic signal, and his idea was to look at the alignment as an intrinsic signal. But there is something else that I have sort of glossed over. And the something else is when my neural network has 3 output layers, 3 output units, HEL. Now, in one case, the example may be 0.5, 0.2, 0.3. And another example, the answer may be 0.8, 0.1, and 0.1. In both cases, I would say the prediction is helix. Because in both cases, helix is the strongest unit. But intuitively, you may immediately argue, wait a minute, this prediction somehow is stronger than that. So here, helix is predicted very clearly as the absolute majority, and nothing else has any sort of significant contribution to the one. While here, helix is 50-50. Okay, it's the strong, we have 3 units, so 50% is the strongest output, but it's still a 50-50 somehow case, right? So this one, they may both be helical residues, but this one is much stronger. So why not use this strength of this prediction to see how well I do? That's what I show. I show the strength of all the residues, and I show here the performance for the 500 proteins. And you see that there is some correlation, okay? If all residues have a higher average strength, you're more likely to be on the right side here of the distribution. If they all have a low number, you're more likely to be on that side. That at least allows you to give with a probability an idea to the user on which side of the distribution are you most likely to sit. It does again not allow you to sort of switch the prediction. It does not, in this particular case, yes, the lowest one is not quite this, so there are others that are low that are predicted well. So it does not allow you to improve the prediction. So if you just took whatever is below a certain threshold to get this one, would we have to be below that threshold, you would in fact turn many that are way better than random into random predictions. So turning everything below a threshold into random would be bad, right? You can see that. So you cannot correct the prediction in this particular case, but at least you can label it. At least you can tell the user, okay, I do not know what to do, but this is less reliable than that. And that's an important thing for people to look at. And I believe at the reliability index, I will get back to that issue on Thursday. And I will close it here and open it for questions to issues we discussed today and issues we discussed before. Questions?